Hey guys, so as I'm sure you are all aware from the shorter to you messages that class tomorrow is a no-go. So that means that I will not be able to go over your study guide with you for next week's midterm. So what I'm going to do is alongside this video link, I'm going to post the study guide. Sorry, I got a little bit of a sore throat, so if you see me drink my tea or take a minute, it's okay. And what I'm going to do is I'm going to go over the study guide with you. And at the end of the video, I'll tell you what to expect on test day. That is a big thing for me. I will give you very clear and very set expectations. So without further ado, if you are coming to the video first, that's fine. But what I would do is go to the document that I have linked in this same module, print off or pull up. It's a PDF, so if you can edit a PDF on your computer, wonderful. If you want to write down the answers and then compare it, or print it off, fill it in, that's the easiest thing to do. But what I'm going to do is I'm going to go over all these answers. You study your study guide, you'll make 100 on the test. Study your study guide, you'll make 100 on the test. There's not going to be anything on the test that's not on the study guide. Not a darn thing. Okay? So, first of all, number 1 on your study guide, actually it's 1 through 5, it says list the steps of the scientific method in order, and each of them is each question 1 through 5 is 2 points. So, number 1, observation. Number 2, you make a hypothesis. Okay. Form a hypothesis about what you've observed. Number 3, you make predictions based on the hypothesis you made. Is it going to work? Is it not going to work? Is it going to do this? Is it going to do that? We used to call that theorizing. They've changed it into making predictions. Number 4, that's when you do your experiment. That's when you start the experimentation. Sorry, my head's hurting. Do your experiment, and experiments should be repeatable. Not that you have to know that, but experiments are repeatable. You can't. It's not just something you can do one time and then we've shot our wad. That's it. Can't do it again. Experimentation. So experimentation number 4. Number 5, draw your conclusions. So come to your conclusions, wrap it all up in a nice bow. Tell me what your hypothesis was, and then tell me what you did in your experiment, and then tell me if your hypothesis was correct. Draw your conclusion, right? Sorry, my computer's shaking. All right. The next one is biology is the blank study of life. It is the scientific study of life, right? We went over that. Next, we're looking at the definition of an endothermic reaction. So just to back up, biology is the scientific study of life. Okay? Scientific. An endothermic reaction is cool to the touch and it absorbs heat. Endothermic. They're like this. They're inside. They don't touch them. They're cold. And then they absorb the heat. They hold on to it. They don't give it off, right? And then on the flip side, an exothermic is going to be the opposite, right? It's hot to the touch and it gives off heat. It radiates heat. It's going to be hot when you touch it because it's giving off that heat, okay? The next 2 are statements that I want you to know. A chemical reaction starts with reactants and ends with new products. And it can't be reversed. Once you chemically change something, you can't, without undergoing another chemical change, you can't reverse the reaction. Whereas a physical change can usually be reversed. Okay? All right. Next, we are looking at blank are examples of, or some examples of blank, basically, are monosaccharides, disaccharides, and polysaccharides. Carbohydrates Monosaccharides, disaccharides, and polysaccharides are carbs. They are carbohydrates, okay? Glucose, the next question. Glucose is a very important monosaccharide. Mono, one. Sucrose, the next question. Sucrose is a very important disaccharide. So, glucose, the G has one hump, so it's mono. Sucrose, the S has two humps, so it's disaccharide. Something to help you remember. All right, next we're going to look at the testing agents that we did in your online lab. Sorry, my arm is tingly, so I'm working it out over here. We're going to look at Benedict's reagents. It tests for sugars. Okay. Biuret's reagent, Biuret's, test for proteins. It's really easy to get Benedict's and Biuret's mixed up. Benedict is sugars. Biuret is proteins. Lugol's or Lugol's iodine is starch. Test for starch. And then Sudan 4, test for fats and lipids. And I want you to know a good food example for each of these. So for protein, an egg. For glucose, just simple sugar. Sugar. Starch, a good starch is a potato. And fat, grease is a good example. There's a multiple choice question asking you about this. So just FYI. Alright. Long chains of amino acids. Those make up what? Those make up proteins. Proteins are made of long chains of amino acids. And those amino acids are held together by a peptide bond. Okay? So long chains of amino acids make up proteins, and amino acids are held together by peptide bonds. And then the next one is another statement that I want you to know. So it's either going to be hydrophilic or hydrophobic, right? So hydrophobic, it repels water. It's afraid, right? You're phobic, you're afraid, you repel. So fats and lipids are hydrophobic. They repel water, okay? They're not hydrophilic, they're hydrophobic. All right. So the next page we're looking at is the page with the chart at the top. So your scanning objective has a 4 times magnification. Your low powered or yellow objective has a 10 times magnification. Your high powered blue objective has a 40 times magnification. Those are all your lenses that are on the nose piece that you turn. Your oculars, which you look through, those are 10. Those are 10 times. So in order to get the total magnification over here for scanning, 4 times 10 is 40. For low powered, 10 times 10 is 100. For high powered, 40 times 10 is 400. So if I were to give you this chart, you would need to be able to fill it in. All of these, okay? All right, next we're looking at diffusion and the fact that it is the movement of particles. And osmosis is the movement of water. Both things move from areas of higher concentration to lower concentration. Diffusion is particles. Osmosis is water. Okay? Next, we're going to look at the tonicity questions. All right. So, an isotonic solution is the one where the concentration of the solute and the solvent inside and outside the cell are the same. So, the concentration of water inside the cell is the same as the concentration of water outside the cell. So, your cell just kind of I'm not telling you that there's no movement, but what I'm telling you is that there is no net movement. So that means there's not one that goes whoop or one that goes whoop, right? It doesn't shrink. It doesn't balloon. It stays. It's in homeostasis. Okay. Next, we're going to look at a hypertonic solution. So hypertonic is the water inside the cell. The concentration of the water inside the cell is greater than the concentration of the water outside the cell. So when the concentration of water is greater inside the cell, it says, "Ooh, the outside is thirsty. It needs water." So it goes. So when that happens, the cell goes from normal, that water leaves that cell, and that cell goes, shrivels up. It's hypertonic. So the net movement is out of the cell. Okay? So on the flip side of that, hypotonic is when the water on the outside, the concentration of the water on the outside is higher than the concentration of the water on the inside. So the water goes from outside the cell into the cell. So that cell goes from being happy where it's at, and it's going to balloon out because all that water's coming into it. So the water moves into the cell on the hypotonic, okay? The two types of cells found on Earth are prokaryotic cells and eukaryotic cells. Okay? So the next thing we're going to look at on your document are the two equations that I put on there. And it's photosynthesis and cellular respiration. And so what I want you to do is I want you to make some notes because I accidentally forgot a page of the study guide and I need you to write this down, okay? So you can pause me because I'm just going to keep going and then pick me right back up when you come back with your paper and pencil if you don't have extra, or you can flip it over onto the back of the document if you printed it out, okay? So what I need you to know is that photosynthesis begins with an inorganic carbon and water, okay? So if you are given this equation where it says photosynthesis, it's going to start with an inorganic carbon. It's typically CO2 gas, right? And water. Now, for photosynthesis to happen, you have to have light and you have to have chlorophyll. If you do not put those, I'm going to ask you to write the photosynthesis equation. This is me giving you a test question. One of the questions is, write the equation for photosynthesis, okay? And I want it balanced. I want the numbers to match. I want them to be what they're supposed to be. You have to put in there, in the middle, on the arrow, light and chlorophyll, or I'm marking some credit off, okay? Has to be that way, okay? And then it starts with an inorganic carbon and water. Light and chlorophyll get introduced, and it produces glucose, water, and oxygen. So, write this down. It starts with an inorganic carbon and water. As a byproduct, the two byproducts that I want you to focus on are water and oxygen. And then the glucose comes into play with cellular respiration. So cellular respiration uses what for energy? Glucose. Look at the equation below. C6H12O6 is glucose plus oxygen gas yields CO2, water, and ATP. So they're not reversible equations, because sometimes with chemistry you can do that, but it's not that way on this one, okay? So it is not reversible. It's close, but it's not. But that CO2 and water gets recycled and back into use for the beginning of photosynthesis. And then that ATP you'll see in a little bit, gets used in the cells for cellular energy. And we'll go over that in just a few minutes, okay? So I want this written down for you, okay? You may have already written some of it down. Photosynthesis begins with an inorganic carbon and water. It has to have light and chlorophyll. Cellular respiration uses glucose for energy. Photosynthesis creates water and oxygen as a byproduct. And in cellular respiration in living cells, carbon dioxide is given off, CO2. And in the lab online, there's going to be a video about showing cellular respiration. Yeast cells are used to show that, okay? All right, yeast cells. All right, so that is it as far as questions go. Actually, nope, I lied. I'm sorry. I also have this, and it is on the document. What I want you to know are these 5 things: the plasma membrane, the smooth endoplasmic reticulum, the nucleus, the Golgi apparatus, and the mitochondria. I want you to know what those look like on this. I want you to be able to label those 5 things. And I want you to give me the function of those 5 things, okay? So the membrane regulates passage of things into and out of the cell, right? It's that first line of defense. It regulates into and out of. The smooth endoplasmic reticulum is involved, what I want you to know is it's involved with the lipid synthesis, right? All right, it's involved in the lipid synthesis. Your nucleus, that's the brain of the cell, right? That's the control center. It directs protein synthesis and cellular reproduction. That's where the DNA is housed. The nucleus is the control center. It's the brain of the cell. The Golgi apparatus is basically the post office of the cell, right? It packages proteins and forms these secretory vesicles that send things out. It packages up and ships it out, right? It's the post office. It's the ups truck. And then the mitochondria, the powerhouse of the cell. I kind of did this ironically because everybody always knows that the mitochondria is the powerhouse of the cell. But why is the mitochondria the powerhouse of the cell? Because it is the site of cellular respiration, which we just went over this with cellular respiration. In cellular respiration, ATP is produced, and ATP is the energy that we use in our cells to go, right? So make sure you know that, and make sure that you can label these 5 on this chart, okay? All right, so that's it as far as the study guide goes. Now, I'm going to give you very clear, very concise, very upfront expectations for my test. I have had something come up on Thursday of next week. So, your lucky day is that your test is going to be online on Canvas. It will open at your lab time. It will open at 5:30 on Thursday. It will close at 7:20 on Thursday. That is a 2-hour window to take your test. It is the exact same time window that we have for lab. I'm not going to give extra time. I'm not going to do anything like that, okay? I'm giving you this heads up. I'm also going to tell you very clear, I understand that I am giving an online midterm. I understand that you will have resources at your fingertips, i.e. your study guide that we just went over. I know that that will be out, and I know that you will use that, okay? I wouldn't expect you not to use that. But I do expect you to study, because if you don't, This test will kick you in your tail if you don't study, and it'll take you too long to take it. It's 50 questions. It's 100 points. 2 points per question. Okay? Canvas will grade most of it for me. There are some that I will go in and grade myself. You will have 10 possible bonus points. Okay? 10 possible bonus points. And those bonus points, in order to get all 10, you got to get all parts of the question right. It's not all parts of, it's more than one question, but in order to get all 10, you got to get it all right. Now, it's not all or nothing. You can get some without getting all of it. But with that being said, email me with questions. Send me a Canvas message if you have any questions. I'm going to post everything right now. It's Wednesday at about 5:10 while I'm recording this. And I'm going to go ahead and get it all posted. I'm going to schedule for your midterm to open. I will make an announcement in Canvas, and I will send out an email with the same information as far as test expectations, test time, test format. Okay? So if you have any questions, please let me know. I'm more than happy to answer anything that I can. And study your study guide. Study your study guide, and the test won't take you long at all. You might not even need it out while you take it. Okay? Godspeed, study, and I will not see y'all until the week after fall break. So, enjoy. Bye.